This conference will now be recorded.
Hello students. So for the NEET exam we are preparing. So here we are going to learn 14th chapter, respiration in plants. We'll just go through the important points of this chapter. So let's see, according to NEET syllabus, what and all you need to learn in this chapter. The main concept is respiration. In that, exchange of gases, then cellular respiration. Cellular respiration concept, in that you are learning about glycolysis, fermentation, anaerobic that is anaerobic respiration, that's fermentation, then TCA cycle, then electron transport system, then energy relations, under that number of ATP molecules generated, then amphibolic pathways, respiratory quotient. About all this, these are the main concepts from where you will get questions in NEET exam. Okay, so let's see one by one. Here first we need to learn what is respiration. So we know that respiration means that is enzymatically controlled catabolic process. Okay, so there are so many catabolic processes which are enzymatically controlled. Okay, so here all these controlled, enzymatically controlled catabolic processes, and these processes, these catabolic processes releases energy. Okay, this releases energy, and these catabolic processes which releases energy, that is by stepwise oxidative breakdown of food substances. So here food substances are getting broken down. So this is oxidative breakdown of food substances. That's how energy is getting released, by oxidative breakdown of food substances. That is happening here. That's how it is getting released. Energy is getting released. Then next point is cellular respiration. Cellular respiration. So let's see, the cellular respiration means that is the stepwise oxidation of food molecules. Okay, stepwise oxidation of food molecules in a cell. In a cell, the stepwise oxidation of food molecules, and that is using oxygen. So it is leading to the production of carbon dioxide, production of carbon dioxide. Then water. Water is also getting produced. Then energy is getting released. Okay, these are the processes. Carbon dioxide is getting produced, water is getting produced, and energy is getting released. So this is the releasing of energy. Here the releasing of energy is taking place, and the energy produced, that energy which is released here, that energy is produced on It is getting stored in phosphate bonds of ATP. Okay, where this energy is getting stored? In the phosphate bonds. Phosphate bonds of ATP. Their energy storage. Okay, these points I'm explaining based on last 10 years question paper, previous 10-year question papers. So from that, each and every point you need to note down, because all these points have come in the previous year question paper. So this class or this chapter notes are completely, all the chapters I'm doing like that only, previous year question papers, and based on that these points are explaining. So here the utilization. See here, first of all, energy is getting produced and then it is getting stored in the phosphate bonds of ATP, and that is getting utilized. This ATP, this energy which is stored, that is getting utilized in the life processes. This energy is getting utilized for the life processes. So ATP, that's why the ATP is called as energy currency of the cell. Okay, energy currency of cell. Okay, so here cellular respiration means stepwise oxidation of food molecules and their oxygen is getting utilized. Then in that stepwise oxidation, what is happening? This is leading to the production of carbon dioxide, water, all these are getting produced along with energy, and energy is also getting released, right? So this produced energy or this released energy will be stored in the phosphate bonds of ATP, and afterwards this energy which is stored, that is getting utilized for the life processes. That's why ATP is called as the energy currency of the cell. Next, respiratory substrates. Here, see, organic substances, the organic substances which is getting oxidized. We said this is an enzymatically controlled reaction, right? So here, substrate should be there, enzyme should be there, that should work, reaction should take place, then the release of energy is taking place, right? So here the respiratory substances are the organic substances, organic substances, and these organic substances are getting oxidized and they are liberating energy during respiration, right? We said what is respiration, the same thing here we are explaining. These organic substances, this is getting oxidized, right? And after oxidation they'll release energy during that respiratory process. Clear? Next, about carbohydrate. Carbohydrate you will learn very clearly in this zoology portion. Carbohydrate, or we can say glucose. Okay, this carbohydrate, they are the most common substrate which is used in the cellular respiration. This is the common substrate for cellular respiration. Okay, and glycogen and the sugars, other forms of sugars other than this glucose, there are some other forms of sugars. This glycogen, these are broken down to glucose and these are consumed in glycolysis. Okay, this is consumed in glycolysis. Okay, and here we need to learn about different types of respiration. Types of respiration, we need to say first about two types of respiration, anaerobic respiration and aerobic respiration. Then second type is aerobic respiration. Here in the anaerobic respiration, first we can say that anaerobic respiration is taking place in the cytoplasm. This is in cytoplasm, which one? Anaerobic respiration. And this will not use molecular oxygen. No molecular oxygen is utilized here. Okay, there is no usage of molecular oxygen. And in anaerobic type of respiration, electrons which are removed from the substrate, electrons which is removed from substrate, all these electrons are getting transferred by oxidoreductase enzymes. This is transferred by oxidoreductase enzyme to where it is getting transferred? To electron acceptors other than oxygen. To electron acceptors other than oxygen. Okay, so this process is called as fermentation. Fermentation. This is called as fermentation. This is the anaerobic respiration. Okay, and this involves in the formation of different products like alcohol, organic acid, carbon dioxide. Here, alcohol, organic acid, then carbon dioxide, all these productions that involves in the anaerobic respiration. Okay, so these are the main points you need to learn under anaerobic respiration. And next comes the aerobic respiration. Here we'll see what is aerobic respiration. You need to differentiate what is aerobic respiration and anaerobic respiration. Here in the aerobic respiration, this leads to the complete oxidation of organic substances. Okay, aerobic respiration, first of all, it is taking place in the presence of oxygen. Okay, so this process, this aerobic respiratory process leads to the complete oxidation of organic substances. Complete oxidation of organic substances. Okay, and here it is in the presence of oxygen, right? And here after this oxidation, that releases carbon dioxide. Carbon dioxide is getting released, then water is getting released, then large amount of energy which is present in the substrate. Here one substrate is getting oxidized, right? So the oxidation of organic substance is taking place, and this is in the presence of oxygen that releases carbon dioxide, water, plus lot of energy which is present in the substrate. Clear? Then aerobic respiration means that includes, this is very important, aerobic respiration includes 5 independent major processes. See, what is aerobic respiration and anaerobic respiration? Here I am mentioning the 5 major processes which are included or which are there, which are making this aerobic respiration. First one is glycolysis. Then pyruvate oxidation. Krebs cycle. Electron transport chain. electron transport chain or ETC. Then oxidative phosphorylation. So all these processes together, that makes the aerobic respiration. Okay, so aerobic respiration consists of 5 independent major processes. These are major processes and these are independent. Okay, so which are they? Glycolysis, pyruvate oxidation, then Krebs cycle, electron transport chain, and oxidative phosphorylation. Clear? These are the steps which makes an aerobic respiration. So here we can differentiate the aerobic respiration and anaerobic respiration and the processes which is including in the aerobic respiration. Okay. Next, first we need to learn about glycolysis. See the one process of aerobic respiration. We said glycolysis, this is taking place in the cytoplasm. Okay, and this involves the reduction of first it is starting from glucose to glucose 6 phosphate, fructose 6 phosphate, fructose 1,6 by phosphate, then here 1,3 bis phosphoglycerate, 3 phosphoglycerate, here 2 phosphoglycerate, then phosphoenol pyruvate, and finally it is forming pyruvate. Okay, so from glucose, pyruvate is getting formed. Next process, another independent process will come, right? Pyruvate oxidation. Then comes Krebs cycle. Then electron transport chain. Then oxidative phosphorylation, right? Here you can see the enzymes which are from glucose to glucose 6 phosphate. How it is forming? Here hexose kinase enzyme is working. So here the major thing is you need to learn all these steps, glucose, glucose 6 phosphate, fructose 6 phosphate. Along with that, you need to mention the enzyme also. Here for NEET exam, you will get the step and which enzyme is working there or what is the product of that reaction. Glucose plus it is in the presence of hexose kinase enzyme, it is forming. You'll get options. You should select the correct option, glucose 6 phosphate, right? Next, glucose 6 phosphate. From that, phosphate glucose, it's almost you can connect the names, phosphate glucose isomerase enzyme. Then it is forming fructose 6 phosphate. Then in the next enzyme here you can see the phosphofructose kinase. So here you can relate that fructose 6 phosphate to fructose kinase. Okay, so like that you need to connect all these and learn. So this occurs, glycolysis is taking place in a cell. Glycolysis is taking place in cytoplasm, and here the reduction of one glucose molecule to 2 molecules of pyruvate. All these steps, finally it is ending one glucose molecule. Here it is starting from one glucose molecule and finally it is forming 2 molecules of pyruvate. 2 molecules of pyruvate, right? So this process that is called as glycolysis, and here in glycolysis you can see here the ATPs. In the whole process, 2 ATP molecule is getting utilized. See, it utilizes 2 ATP molecules. Here you can see ATP is getting converted. Here another step, ATP is also getting converted here. In the other places and all, there is no ATP is getting utilized. So here 2 molecules of ATP that is getting utilized and that produces 4 ATP molecule Here you can see 4 ATP, here 2 ATP, here 2 ATP. Total 4 ATPs. 4 ATP molecules are getting produced. And 2 NADH. Here you can see 2 NADH is getting produced. This 2 NADH that is getting produced. So the net gain of ATP in glycolysis, that is 8 ATP molecules. Net gain. These are the important questions. 2 ATP molecules are getting utilized, 4 ATP molecules are getting produced, 2 NADH is getting produced. So the net gain, we can say net gain of ATP in glycolysis, that is 8 ATP molecules. Okay, net gain of ATP molecules in glycolysis is 8 ATP molecules. Clear? Here, glycolysis is common to both aerobic and anaerobic respiration. We have learned first we were explaining about aerobic respiration and anaerobic respiration. So glycolysis is the common process. Glycolysis is common for aerobic respiration and anaerobic respiration. Glycolysis is the common for aerobic and anaerobic respiration. See, in the muscles of humans during the exercise or exercise time, what is happening there in human muscles? Pyruvic acid is reduced to lactic acid during exercise. Pyruvic acid that is getting reduced to lactic acid. It's forming lactic acid during exercise time. Okay, and there one enzyme is working, that is lactate dehydrogenase. Here which enzyme is involved? This is one question, one important question, repeated question. Here the enzyme is lactate dehydrogenase. Lactate dehydrogenase. This enzyme is working and it is reducing the pyruvic acid to lactic acid during the exercise. Muscles of human during exercise, pyruvic acid is getting reduced to lactic acid in the presence of which enzyme? Lactate dehydrogenase. And in fermentation, which is an anaerobic respiration. So in fermentation by yeast, here pyruvic acid is getting converted to ethanol and carbon dioxide. Pyruvic acid is getting converted into ethanol and carbon dioxide, right? And here which enzyme is there? Pyruvic acid decarboxylase enzyme. Which enzyme is present here? Pyruvic acid decarboxylase. Pyruvic acid decarboxylase enzyme is present, and one more, alcohol dehydrogenase enzyme. Alcohol dehydrogenase. So these enzymes are present during this process. Okay, fermentation. In that, this is an anaerobic process, fermentation. So pyruvic acid is getting converted into ethanol and carbon dioxide, and the presence of two enzymes we can learn here, that is pyruvic acid dehydrogenase, because pyruvic acid is getting converted, right? So this enzyme is easy to remember, pyruvic acid dehydrogenase, and here ethanol is getting produced. So alcohol dehydrogenase, that is also there. Ethanol, that is alcohol. So ethanol dehydrogenase. alcohol dehydrogenase is present here, the enzyme. Clear? Next point which we need to learn here is so here about glycolysis, we have to learn the whole process, stepwise process, these names and the enzymes which are involved and total ATP gain and utilization of ATP, total number of ATP, then net gain of ATP you need to learn. Then this glycolysis, which is the common step for anaerobic and aerobic respiration. And here, what is happening, which and all the enzymes involved. These are the main points from the previous year question papers.
And here you need to learn about Krebs cycle. Next, in Krebs cycle, what you need to learn is pyruvate oxidation will learn. See, pyruvate oxidation, this is taking place in the mitochondrial matrix. In the mitochondrial matrix. Okay. See here in the Krebs, in the glycolysis, you have seen pyruvate is getting produced. Glycolysis, there, pyruvate is getting produced. Right? And this pyruvate enters to the matrix, mitochondrial matrix. This enters to mitochondrial matrix. How it is entering? This entry is with the help of some specific transport protein. With the help of transport protein. Some protein, transport protein is helping to enter this pyruvate to the mitochondrial matrix. So while moving like that, that involves the conversion. Here one conversion process is taking place. Conversion of glycolysis product. Which is the glycolysis product? Here, conversion of glycolysis product. That is pyruvate, right? So pyruvate is getting converted. That means pyruvate is getting converted into two molecules of acetyl coenzyme A. Okay, pyruvate is getting converted into two molecules of acetyl coenzyme A. Okay, here you can see glucose that is forming pyruvate. In which one? In the first one, in the glycolysis, right? Then that pyruvate should enter to mitochondrial matrix. That product, that is one independent process. If it is moving to the next one, this glucose is getting converted, or the pyruvate is getting converted into two molecules of acetyl coenzyme A, and the entry to the mitochondrial matrix, that is taking place with the help of transport protein. Okay, it is with the help of that transport protein, it is entering to the mitochondrial matrix, and there one conversion of glycolysis product or the pyruvate is taking place. Which is the glycolysis product? Pyruvate. So that pyruvate is getting converted into two molecules of acetyl coenzyme A. Here you can see acetyl coenzyme A is entering. So that's the entry process. Okay, after this entry, the next process is starting, or the next step is starting, which is called as this acetyl coenzyme A, that starts the initiation, or that gives the initiation step of Krebs cycle. Krebs cycle. Here you can see that acetyl coenzyme A is starting step of Krebs cycle. So this is also called as this change, that is also called as link reaction. This is called as link reaction. This is also one important previous year question, repeated question. Link reaction, because acetyl coenzyme A links glycolysis with Krebs cycle, right? This acetyl coenzyme A, that is the link between the glycolysis and the Krebs cycle. Formation of acetyl coenzyme A, this forms the link reaction between glycolysis and Krebs cycle. So here we need to write pyruvate. Pyruvate, this is a 3-carbon compound. Okay, pyruvate plus NAD plus acetyl coenzyme A. In the presence of which enzyme? Pyruvate dehydrogenase. Pyruvate dehydrogenase. That forms here coenzyme A in the presence plus coenzyme A in the presence of enzyme pyruvate dehydrogenase, that forms acetyl coenzyme A, which is a 2-carbon compound, plus NADH plus H plus carbon dioxide. Here carbon dioxide is getting released. Okay, so this is the summary of the reaction. Here also in Krebs cycle also we said it is occurring in the mitochondrial matrix. Krebs cycle is also taking place in the mitochondrial matrix. Okay, Krebs cycle is taking place in the mitochondrial matrix. So here the acetyl coenzyme A is getting formed, or it is getting utilized, and finally it is forming oxaloacetate. Okay, see from here reaction is starting, acetyl coenzyme A that is working, then finally it is forming oxaloacetate. So this Krebs cycle or the cyclic process that involves the conversion of, this is a cyclic process. So here conversion of acetyl coenzyme A to which one? Oxaloacetate. Oxaloacetate is taking place. Okay, here also stepwise all this you need to remember. Citrate, isocitrate, alpha ketoglutarate, succinyl coenzyme A, here the succinate, fumarate, malate, oxaloacetate. And all these enzymes, if possible, you should remember. Okay, try to remember. That's what these are: glycolysis, Krebs cycle, all these things daily 1 or 2 times you just read so that you will get an idea of next step. Okay, so that produces carbon dioxide. Okay, produces carbon dioxide, which is one of the end product of respiration. Then GTP and reduced coenzymes, that means NADH plus H plus and FADH. So it is producing carbon dioxide. Then GTP, reduced coenzymes, all these are getting produced. And here in the Krebs cycle, total of 24 ATP molecules are getting produced from one acetyl coenzyme A. 24 ATP molecules are getting produced. This ATP is the energy currency, right? Energy currency of the cell, or the stored energy form in a cell. So here 24 ATPs are getting produced from one acetyl coenzyme A. Acetyl coenzyme A molecule. And here net equation, how we can write a Krebs cycle equation. Final equation, how we can write 2 acetyl coenzyme A plus 6H2O plus 6NAD plus 2FAD plus 2GDP plus gives 4 carbon dioxide, 2 water molecules, then 6 NADH plus 6H plus 2 FADH2 plus GTP plus 2 COA. Okay. So this is the final step. But here the enzymes and the products after each reaction that you need to learn. Here the net gain, here the very important point, the total 24 ATP molecules are getting produced from one acetyl coenzyme A. You have to learn that. You should keep in mind after each reaction how many ATPs are getting produced, what and all getting utilized there. Okay. Next comes the pentose phosphate pathway. So now we have covered all the important points of Krebs cycle. Pentose phosphate pathway. This is the alternative to TCA cycle. Okay, this is alternative to TCA cycle, and this occurs both in plants and animals. In both plants and animals, it is taking place. Okay, and here the oxidation of glucose 6-phosphate to pentose phosphate in the cytoplasm is taking place. Cytoplasm oxidation of glucose 6-phosphate to pentose phosphate. Okay, so in the cytoplasm it is taking place. That's the pentose phosphate pathway. Next we need to learn, this is a simple matter. Here you can learn only these 2 points which were in the previous year question papers. And next we are learning about electron transport chain. Electron transport chain, etc. Okay, electron transport chain, where it is taking place, this occurs in the inner mitochondrial membrane. Inner mitochondrial membrane, their electron transport chain is there. Okay, because it contains several protons and electron acceptors. This is having several H plus proton and E minus electron acceptors. So this is having electron transport chain is having, or it is containing several proton and electron acceptors. And in electron transport chain process, electrons passes from one carrier to another. Here electron passes from one carrier to another one. So there, while carrying like that, it produces ATP from ADP and inorganic phosphate. Here ATP is getting produced from ADP and inorganic phosphate. Right. So here the major events which is taking place in the electron transport chain, that is regeneration of the reduced coenzymes for reuse. Regeneration of reduced coenzymes for reuse, right? Then transport of electron to an ordered sequence. Transport of electrons in ordered sequence, in an orderly manner, that transport of electrons that is taking place to molecular oxygen to produce water. Molecular oxygen or O2 form. For which one? For the production of water. Okay, and simultaneously transfer of this produced H plus ions from the matrix to the intermembrane space, which is giving rise to that proton gradient. And that's how the membrane is becoming electric potential. Electric potential of membrane is getting generated. Right. So transfer of protons from the matrix to the intermembrane space, which is giving rise to proton gradient and the membrane electric potential. Then electron transport chain involves 5 complexes. See, electron passes from one carrier to another through these 5 complexes, or from first to fourth complex, and these electrons which is coupled with the complex, fifth complex, all the electrons are getting coupled with that to generate the ATP from ADP. Okay, from first complex to fourth, from first complex to fourth complex, electrons are passing. Okay, from one carrier to another carrier through complexes, electrons are passing. And finally, in the fifth complex, it is getting these electrons, which is coupled with the fifth complex. Here, electrons are getting passed and it is reaching the fifth complex. So, these electrons will get coupled with the fifth complex and that forms the ATP from ADP and PI. Okay, so various electron carriers which is present in the electron transport chain. Which are the electron carriers? Flavins, electron carriers which are present in the, this is very important, flavin is the electron carrier which is present in electron transport chain. Then ubiquinone, then cytochromes, oxygen. All these are the electron carriers which are present in the electron transport chain. Okay, then the complexes we said, the 5 complexes. First one is NADH dehydrogenase. Then another one that is succinate coenzyme Q reductase. Succinate coenzyme Q reductase. Third one is cytochrome C reductase. Fourth one is cytochrome C oxidase. Okay, so these are the complexes. 4 complexes, and the fifth one we said all these electrons which are formed that will couple with the complex 5 to generate ATP, right? Next, we need to learn about, these names are important. Next, we need to learn about oxidative phosphorylation. From this topic also, you will get repeated questions. So here in the oxidative phosphorylation, that involves the synthesis of ATP or synthesis of ATP molecules from energy liberated during electron transport chain. Energy which is liberated during electron transport chain. And here enzyme which is used. Which enzyme is used? ATP synthase. ATP synthase enzyme is getting utilized, or that is getting utilized for the production of ATP molecule from the energy which is liberated during electron transport chain. And this enzyme that is found in the F1 region of the elementary particle. Where this enzyme is present? Enzyme ATP synthase. That is present in the F1 region. In the F1 region of elementary particle which is present in the mitochondrial membrane. F1 region of elementary particle of mitochondrial membrane. Okay, this is very, very important, repeated question and slightly confusing, right? So enzyme ATP synthase will be present in the F1 region of the elementary particle of mitochondrial, which is present in the mitochondrial membrane. Okay, and here, according to the chemiosmotic hypothesis, according to this, here H plus ion translocation is happening. So this H plus ion translocation, that creates a pH difference and membrane potential. pH difference and membrane potential this makes, because H plus ions are getting translocated. So that translocation of H plus ions creates pH difference and membrane potential, and that forms a protein motive force. This pH difference that causes proton motive force, right? That creates a proton motive force, and that causes flow of protons from the intermembrane space down their electrochemical gradient or down to the matrix. So the protons that will pass through the channel in the F0, F1 ATPase particle. Okay, so through that proton travels. Okay, here the respiratory balance sheet we need to learn, which is very important, and you can expect one question from that. Respiratory balance sheet. So in glycolysis, ATP, these are the processes, and here ATP formed, I'll write, and a number of NADH and FADH2. Glycolysis, in that how many ATPs are formed? 2, right? And number of NADH and FADH2, 2 NADH. 2 NADH is equal to 6 ATP. Okay, here 2 molecules of NADH is getting formed during glycolysis. So this is equal to 6 ATP molecules. So here in total, you can see glycolysis, 2 ATP is formed and 2 NADH, which is equal to 6 ATPs. Next, the formation of acetyl coenzyme A. Formation, right? In that, there is no ATP, but 2 NADH molecules. Again, 2 NADH is getting produced, which is equal to 6 ATP, right? And the third one, Krebs cycle. Krebs cycle. In Krebs cycle, you can see 2 ATP, right? And there is 2 FADH2. That means it is equal to 2 ATP and 6 NADH. Okay. 6 NADH, that is equal to 18 ATP. This is for which one? Krebs cycle, right? So total, you can see here, total 2 plus 2. This is 4 ATP, and here 36 or 32 ATP. That in detail you will get to know. 34 ATPs. Okay. So the net gain of ATP during aerobic respiration of one glucose molecule is 32 plus 4. Net gain. That is 32 plus 4, which is equal to 36. Okay. Here, one glucose molecule. Net gain of ATP during the aerobic respiration of one glucose molecule, that is 36. Okay. Next, we need to learn about, so this balance sheet you need to remember where and all, how many ATPs are getting produced. Then amphibolic pathway. Here in the amphibolic pathway, respiratory pathway, which is involving both anabolism and catabolism. Pathway which includes anabolism plus catabolism. Okay, so that's why it is called as amphibolic pathway. Anabolism and catabolism both are involved in this respiratory pathway. So all carbohydrates are getting converted into glucose before being utilized in respiration. All carbohydrates. All carbohydrates are getting converted into glucose before being used for respiration. So carbohydrate is getting converted into glucose. And fats, that will get broken down into glycerol and fatty acid. Fat to glycerol and fatty acid. This is further converted into acetyl coenzyme A, and then it is entering to the which one? Respiratory pathway. This later, this will get converted into acetyl coenzyme A, then to respiratory pathway it is entering. Okay, there is a connection between all these things. Then protein, that is broken down into amino acid, and this is again entering to the Krebs cycle. Protein, this is broken down into amino acid. Again, that is entering to the Krebs cycle. Okay, so this is how the anabolic pathway you need to explain. Then the respiratory quotient, RQ. Respiratory quotient. So respiratory quotient means the ratio of volume of carbon dioxide which is evolved to the volume of oxygen which is consumed during respiration. So oxygen is consumed and there is a ratio between oxygen consumption or the oxygen which is consumed during respiration and the volume of carbon dioxide evolved during that respiration. So this is depending upon the type of respiratory substrate, and here the respiratory quotient value that varies. For carbohydrate, respiratory quotient is 1. Organic acid, for that more than 1. Respiratory quotient will be more than 1. Okay, and fat or protein, for that respiratory quotient will be less than 1. Okay, then succulent. For succulents, respiratory quotient is 0. So this also you need to remember this. In so many question papers, this value, respiratory quotient values they have asked. It's a direct question, just you need to by heart these values. That's all. So here, all the exams, for all the exam questions of this chapter that I have discussed with you under these 4 topics like glycolysis, Krebs cycle and electron transport chain, then the respiratory quotient and respiratory balance sheet, then fermentation, all these things. Under all these things, we have discussed almost previous year 10-year question paper. From the question paper, we have taken all the questions and discussed. Next, you need to make a note or cover all these topics. Just go through all these topics and answer previous year question paper. Just try to understand or try to make out how many questions you can answer from this after reading this note. I've covered almost all the questions.
So read all these portions very well, and the next class will go to the next chapter. Thank you.